সেখানটা চলো, সেখানটা চলো, আসছে না, আসছে না, এটা মাইক ক্লিন নেবে না, একটা মাইক বন্ধ
Hello everyone.
Hello, sir.
So Prasenjit, you are conducting.
Yes, I am conducting the Google Meet, and my colleague Dr. Shilajit Borua is conducting the YouTube live streaming.
Yes, Dr. Champa Devnath.
Welcome,
Thank
Shumita.
you. Thank you. So my slides are ready. You just tell me when to start.
Okay, before starting, there will be a short introduction, and within 5 to 10 minutes, it will start. Okay.
Sure. Thank you.
Uh, so, Shampadi, we will wait for 1 more minute, and then at just 7, we will start. Okay.
Okay. Okay.
Anchors, are you ready?
Yes, sir.
Okay.
Yes, sir.
Uh, I'll start the recording at first, then anchors, you will start the program. Okay. So it's dot 7 p.m. and we are going to start. Let me start the recording first. Okay, let's start.
Good evening to one and all present here. It's my pleasure to welcome everyone to today's biology lecture series. I, Anandita Paul, will be hosting this webinar along with Shantari Sen Sharma. On the behalf of the institution, VijaygarhJyotishRay College, I would like to extend a special welcome to our guest speaker, Dr. Samir Kumar Patra, Professor of the National Institute of Technology, Rourkela, who will be speaking on the topic Molecular Logics of Life, Chromatin Modifications and Gene Expression. Thank you.
Thank you, Anandita. Now, I would like to request the head of the Department of Microbiology and the IQAC coordinator, Dr. Proshenjit Das, to welcome our honorable guest speaker, Dr. Samir Kumar Patra, the convener, Dr. Shampa Devnath, and other respected teachers and students to join us. Hope today's discussion will inspire ideas among the inquisitive minds.
Thank you, Shanchari. This is the third session of the biology lecture series that we had started in the first week of September this year. And in this lockdown period, these sessions are the best way to utilize and to nourish our knowledge base. And with us today, here is Professor Samir Kumar Patra from Department of Life Science, National Institute of Technology, Rourkela. I welcome Dr. Samir Kumar Patra with all my heartfelt gratitude. Uh, I guess this lecture is going to help the student to learn their topics of molecular biology, because I guess this chromatin modification and gene expression, this is a very important part of their syllabus and for their future endeavor in research field very soon. So students are also welcome here. I request all the students to enjoy the lecture session and put your queries, put your questions in the chat box so that this webinar will be more interactive, more interesting, and more enjoyable. With this brief welcome note, uh, the anchors, you are requested to proceed with the next part of the program. Thank you.
Thank you for your speech, sir. Next, the convener of the webinar, Dr. Shampa Devnath, will give us a brief introduction about today's guest speaker. Thank you.
Thank you, Anindita. Dr. Samir Kumar Patra is currently Professor of Biochemistry and Molecular Biology at the Department of Life Science, National Institute of Technology, Rourkela. Professor Patra was awarded Ph.D. degree in 1997 under the supervision of Professor Mednikanta Pal. After that, he did his research work as postdoctoral fellow in the University of Basque Country, Bilbao, Spain during 1997 to 1999, in the University of California at San Francisco during 1999 to 2002, and in Canada during 2002 to 2004. He also did his research work under the mentorship of famous epigeneticist Moshe Zaif, and returning to India, he joined GRF Kalyani and also served as visiting lecturer at the Department of Biotechnology, University of Burdwan. In July 2009, he joined at the Department of Life Science, NIT Rourkela, and developed the Life Science Department from the beginning to its current international status. Professor Patra's major scientific contributions are mimic demonstration of the existence of bilirubin as liquid crystal in circulation during jaundice, nature of bilirubin binding to serum albumin, explored in the expression, assay and overall role of DNA methyl transferases, histone deacetylase, delineation of membrane signaling hubs as cholesterol raft and ceramide raft, demonstration of epigenetic drift toward histone modification over DNA methylation in regulation of gene expression by epigenetic modulation, embodiment of DNA demethylation mechanism without breaking the DNA in gene expression during development in aging and cancer. He has published well over 70 research and review articles. Professor Patra is the editorial board member of BBA Molecular Basis of Disease, Elsevier, Environmental Epigenetics, Oxford Academic, and associate editor of Biochemistry and Biophysics Report. Thank you, Anindita. You may proceed for further. Hello, anchors. Are you here?
Yes, ma'am.
You may proceed. Huh?
Sanchari, you may proceed. I guess Anindita has got disconnected or some technical problem crop up. So proceed with your next part.
Sir, the next part is where the Dr. Samir Kumar Patra is going to speak now.
Okay.
You request
Okay.
the speaker.
Okay, thank you. Prasenjit, I don't know you personally, but I think you are junior to Sampad, that's why I call you by name.
Absolutely okay. Not a problem.
Okay, so thank you very much.
Thank you.
Sampa also. Sampa is well known to me since her MSc studies. She was just 2 years junior to me in the Department of Biochemistry and Biophysics, Tulane University. Thereafter, we did Ph.D. in the same department for some times and in the Tulane University. So we know each other, and thanks to the social media, we are again connected. Because otherwise, in the middle time, we were not known who is where. So, thanking to the principal of this college, I am starting my lecture today. So I am sharing the screen. Let me share the screen.
I am stopping my presentation.
Yes. No. Acha. So if I share a window, that will be okay, or I will have to share the entire screen?
No, a window is sufficient.
Okay.
Yes, your screen is visible.
Okay, every full screen okay for everyone?
Yes, okay.
Okay. So I again thank to the principal and Sampa and Prasenjit for inviting me and giving me the opportunity to explore some of my knowledge which I have gathered during the last 35 years of my study in science, particularly chemistry, biochemistry, as well as epigenetics and gene expression pattern. So since Sampa told me there will be from first year to MSc students and some of the lecturers and college professors and interdepartmental students as well as professors. So I started and prepared my lecture in this way. So what is life? Particularly if we want to learn what is life, life can be best described with some of the patterns that follow by living organisms. And that was described in 2000, nicely described in 2002 by great scientists. You know all of them by those who have studied the enzyme activation, enzyme kinetics, induced fit model. Can anyone tell who is that scientist who put forward the induced fit model? Any student?
Sir, Coastland.
Yes, good, very good. So the Coastland has proposed in 2002, and that appeared in two-page paper in Science. But after the end of his career, his editorial capabilities, teaching, research, what he thought, he explored that one, and that we and all the scientific community, particularly I love that one. That is 7 pillars of life. What is that? You can write, those who want to learn, that PICERAS. P is the programming. All the living organisms, life process is programmed, and that is programmed in DNA. Clear? That is nucleic acids, DNA. Particularly, you can argue why not RNA. RNA, particularly some RNA viruses are there, but from the logical point of view, they cannot replicate instead of a suitable host. So we will stick our definition that nucleic acid, particularly DNA, that is programmed. Every organism's life is programmed on that. And how at the end of the lecture you will clear that yes, it is programmed by that way. Then I is improvisation. What your program is there, but how to say it, where to say it, you always need and improvisation to apply it. Then if we consider a living cell, the compartmentalization is there, PIC, compartmentalization. Then the term is E, that is energy. In all biological processes, energy is required, and this energy, glycolysis is the ultimate source, and then you will from protein, etc., through TCA cycle, tricarboxylic acid cycle, and various other in mitochondria, you will find that ATP is formed, the universal currency of energy in living organisms. PICE energy हो गया, RAS, R is regeneration. Every living organism has the capability to regenerate. And another then is adaptation, PICERA. Adaptation is essential in according to the environment, according to the evolutionary pattern. If you analyze, you will see that yes, it is essential. And then last term is S, that is seclusion. It is not least. Seclusion is essential. A reaction is ongoing in compartments, but if you don't stop, it cannot. If it goes entirely through its life process, then it will harm the body. So a certain reaction is essential that will happen, and it will be stopped again. That is seclusion. So in this way, if you think that life perhaps started on Earth 3.5 billion years ago. So, and the Urey-Miller experiment, the sparking, particularly the elemental compositions and electrical spark, and life without the E. coli, initial biochemistry, research, etc., you cannot think. And this E. coli bacteria normally live in our intestine of healthy people and animals. Most types of E. coli are harmless or cause a relatively brief diarrhea, not too much. However, strains like E. coli 015H7, it causes severe stomach cramps, bloody diarrhea, and vomiting. Then if you think another one, Salmonella infection. Salmonella is also, it remains in our intestine, but it is a foodborne illness. But when there is too much, and if you take the meat from that infected animal, then you are contaminated. And when Salmonella bacteria infect, it causes gastrointestinal symptoms, including diarrhea, abdominal pain, nausea, and vomiting. So you read too much about your microbiology in your classes. Now I will come to what life started in water. So what is water? Any student?
Sir, please repeat the question.
Huh?
Please repeat the question.
What is water?
Sir, polar
Jol
solvent.
ki.
Polar solvent.
Huh?
Polar solvent.
Molecular nature bolo.
Sir, polar.
अरे मोलिकुलर नेचर तो पोलर है, पोलर तो can you see the screen?
Yes, sir.
It is written what is water. This is dihydrogen monoxide. If you say H2O, is H2O water?
Yes, sir.
No, sir. H2O is not water. H2O is the water molecule, a simplest component, but single H2O cannot give you water. So, n number of H2O molecules, when they are hydrogen bonded and makes a liquid structure, that is known as water. That we know from scientific calculation. You see, these are the hydrogen bonds, and if you look into the tetrahedral distributions of oxygen, the bonds, here the lone pairs are there. This is the 2, this is 2, and 2 lone pairs are there. So octet rule follows. So that is the structure of H2O, and now they are forming different hydrogen bonds, and then you are getting a liquid form. Now if you look into the river water, pond water, etc., you will see a lot of mixings of different contaminants are there. If you purify and put them there in different states, they will restructure, and you will get different. You see here, this is one type, the restructure, the structure of water. A lot of research is ongoing nowadays. And another issue, another thing is that when hydrogen bonds, hydrogen bond length is flexible, and it can bend also. So depending on the distance of hydrogen bond, if it is little tighty, then the water is more compact, and it generally happens at the 4 degrees centigrade. So that's why water is heavy at 4 degrees centigrade and it is more compact. So similarly, depending on the temperature, it varies also. So that's why water is an miraculous solvent, and it is solvent for life. Now coming to the point, what is chemistry? Carbon, hydrogen, nitrogen, oxygen, various metals, the chemist study. Then catalyst will come, otherwise no chemical reaction will be faster and the chemist will be bored. So water molecule is always essential, for particularly when we study about the living organism materials. If we go for different organic solvents and others for organic purposes and reactions, these are different, but that is not, beyond today's discussion. We will not discuss when we will discuss about the life processes. What is the most important and universal solvent for life processes? Now, if we think,
sir, your slide is not changing, I guess.
Where it is, you read it. Where what you see, you read.
Just first slide. The
My
first
God.
slide is there.
Can you see now?
No, still not now.
Just a minute. A window. Share. Can you see?
Yes. The second slide is visible. Yes. Or the fourth slide. So, sorry, fourth slide. Yes, now fourth slide is visible.
Okay, but why not first slide? Now first slide, okay?
Yes, first slide.
So what I talk about this, can
Now,
you see this
yes,
one?
Yes, now.
So
this is
what
it.
I talk about E. coli and Salmonella, this is the schematic diagram of that. Then I was discussing about water structure. What is water? If you look into the structure, H2O, generally the chemist will tell you this is dihydrogen monoxide. Dihydrogen monoxide. So n number of H2O forms the water. Depending on these are the hydrogen bonds, these are the hydrogens, and depending on the intensity, tetrahedral angle of oxygen, distribution of tetrahedral angles of octet, that is electrons in the outer orbital of oxygen. So HO bond, there are 2 electrons, HO bond, there are 2 electrons, other remain as lone pairs. So they can take part in hydrogen bonding. You know that part. Since oxygen is more electronegative, so it will pull little bit more electrons towards it. So as a result, hydrogen will be positive, and in that way, hydrogen bond formation, water structure, as I said. Now, next one is, can you see this one clearly?
Yes, yes.
So chemists will think about all these different kinds of materials, oxygen, nitrogen, iron, etc., and different kinds of carbon compounds, etc., in this way, which are mostly available either in living organism or they can synthesize. Now, those who are reading biology, whether it is microbiology, zoology, botany, when you will come to the laboratory and think about life processes, you have to think about pH. And what is pH? Students will always say, sir, negative logarithm of hydrogen ion concentration. That is okay, we measure in that way. But pH is a scale which says to us that how much acidity or basicity is there in a solution. So some of your students, those who are microbiology, soil microbiology, you are doing, you have gotten a job in the government as a chemist or a microbiologist to test the soils. Then the farmers are there, they will ask, kya hua babu, amar jomite ki hoyeche, তুমি বলবে নেগেটিভ লগারিদম হাইড্রোজেন আয়ন কনসেন্ট্রেশন बढ़ गया or कम हो गया. That will not give them the perfect note. So you will tell that acid base, either acidic your soil is basic, and how it is you can tell that litmus test करके you can say litmus test করে তুমি বলতে পারো যে yes the pH change হয়েছে. So little bit knowledge about that. You know all these Kw is ion product of water. I will not go at depth for these things. You know all. So keeping all these things in mind. Why the scale is 1 to 14 for pH? That is ion product of water is constant. That is this one. That's why it will not go beyond 14. The scale do not exceeds. So if H plus is more than OH, then acidic. If H plus is less than OH, then basic, etc. You know all these things. I am passing quickly for this. You can measure in this way. Now life science and biology. So already life, definition of life, what I love the definition I told you. So study of living organism composed of cells. So what is the difference between life science and biology? Basically no difference. Life science we generally deals with the life processes, but all the molecular compositions are there. And biology same, in most cases, or the dead substance available from the biological living organisms. Like proteins, those who are studying protein we call them biology. Those who are studying carbohydrates we call them biologists. Okay, but when they are matching it with the life processes, all these things, then we call that yes, they are doing complete life science. That is basically no difference. But if you want to remark a difference, that is the difference in my knowledge what I have gathered during the last 35 years. So cells are composed of elements, molecules and macromolecules. So as a chemist, you will see. So if I go to the full screen, is it okay?
Yes, sir.
So you are seeing life and biology, chemist and biologist. You are seeing? Yes or no?
Yes, sir.
Okay. No, actually Google is not functioning with this one. Animation is not possible.
Sir, if you go for presentation mode, then you can go for animation also.
Google Meet. So I am stopping here. Then you tell me how to load it. So share a window. Okay. Then
No, in the presentation mode, in the PowerPoint slide you are showing, you have to go in the presentation mode.
okay.
Then probably the animation will work.
No, nothing is coming here in that. Okay. So you see the screen clearly visible. Is it okay?
Screen is visible.
This screen is clearly I mean, you can see.
Yes.
Yes, yes.
Okay. So, so chemist viewpoint already discussed, and now biochemist viewpoint is the chemistry of living organisms. Now, if you look at the level 1 elements of life: carbon, hydrogen, sulfur, nitrogen, phosphorus, oxygen, and without magnesium and iron, we cannot think about life, because energy for life is stored with the help of chlorophyll and the elemental composition, whatever is there. But iron is, magnesium is also essential. And here in case of any circulatory organism like human, etc., those who have circulation, and blood is there, in the hemoglobin, iron is essential. Many enzyme functions, magnesium ion, calcium ion, etc., is essential. So elemental composition, these are the main, and others are also important. Main is carbon, hydrogen, sulfur, nitrogen, phosphorus. and oxygen. Then level 2, if you come to the molecular units, you will see nucleotides, amino acids, cholesterol, and sugars, hexose sugar, pentose sugars. Then when they are clubbing together, we are getting enzymes, different kinds. And in the level 3, macromolecules are formed with the help of enzymes from these monomeric units to polymeric units. You see nucleotides are here with the help of enzyme. You know DNA polymerase is the machine, and you are getting DNA. Here amino acids are there. Ribosome is the machine. You are getting protein. And then cholesterol. Here you are getting different kinds with the help of enzymes, different derivatives of cholesterol. And with from the sugars you are getting, say, cellulose, cell wall. And then these macromolecules, they are forming supramolecular complexes. Chromosome, you know, then plasma membrane, you see membrane, lipid and protein interactions, and cell wall. So in the then 5th level, we are thinking about a complete cell and its organelles form within the cells. And at the 7th level, we are coming to different organ systems with multi-organ living organisms, plant or animals, whatever be. Is it clear? Now you all know about these cell organelles. Yes or no? Students,
Yes, sir.
clearly give you one important lesson from here. You know all these nucleolus, nucleus, mitochondria, ribosome, endoplasmic reticulum. But when you think about cell division, you know cell division, you are getting from when cells are not, in a tissue. If you think about, say, lung, liver, or any tissue, take a cell. Cells are generally in the G0 stage, resting phase, in a completely developed organism. Different cells in every components of tissue, they are in the resting phase. We call it GG. Let's say in your screen there is a scratch. A cells needs to be divided to fill the gap. So cells will be activated there. So it comes from G0 to G1. That is gap 1 we call. It will gather all the nutrients, etc. Then it will enter into the synthetic phase where all the genetic material, DNA, will be duplicated and various others. Most of the organelles, they will be also as much as possible, not totally duplicated, but as much as possible when they will divide. And then it is coming to the gap 2 phase. It will preparation, the final phase, whether checkpoints are there, whether all the materials are ready or not, and then it will jump to the mitotic division. It is very quick process. Entire cell cycle, if you consider that 24 hours in general, so out of 24 hours, 11 hours is gap 1, then 7 hours is synthetic phase, that is S phase or stationary phase, and then next 7 plus 11 is 18, then 18 plus perhaps it is 5 hours, G2 gap phase. So 23, and 1 hour is for mitosis. Now if you look in mitosis, it will be dissolved, totally dissolved. Your nuclear membrane will be dissolved. Now how it is forming again, a lot of research is ongoing. And there is a, how many of you know about liquid phase transition? Liquid, it is liquid but at different phase. And it happens within the cellular system due to different composition, but mostly the hydrogen bond pattern when changed. I started with the hydrogen bond, shorter hydrogen bond, longer hydrogen bond, bended hydrogen bond. Initially we had the idea that hydrogen bond this little bit distance, as much as possible closer, otherwise it will not form. Yes, that is right. Now different versions of hydrogen bond, and along with that, when the reactions are ongoing within the nucleus during the division, then particularly the hydrogen bond length differs, and differing the total composition, differing the total elemental composition, macromolecular composition, it differs. Then at the dissolved state, where there is no nuclear membrane, then again it will come when the end of the mitosis, again the nuclear membrane will be formed. The nuclear membrane is supplied by whom? Can anyone tell the students? You know endoplasmic reticulum. Endoplasmic reticulum, that is totally a membrane system, and that nuclear envelope is supplied, that is inner membrane, outer membrane system of the nucleus, that is supplied by endoplasmic reticulum. So when initially when it is dissolving, it comes into the form of endoplasmic reticulum. Right? Regarding endoplasmic reticulum, all of you know the smooth and rough. When ribosome binds, it is rough, and when there is no ribosome, that is known as smooth endoplasmic reticulum. I will not go further. And Golgi, you know, these are after protein synthesis. Golgi is known as the post office of the cell. With different kinds of proteins needs to be sorted. So which protein will go to which place, that is generally dictated by Golgi network. That's why it is also known as the post office of the cell. Then typical animal cell, you know, you can draw. Typical plant cell, you know. Now if we come to the animal cell, because I am working with cancer, mainly human tissue samples, cell lines, etc. So I will give you the glimpses of animal cell lines, plasma membrane, then cytoplasm or cytosol, and nucleus. Then in the cytoplasm or cytosol, you will find different cell organelles, then hyaloplasm and cytoskeleton. Cytoskeletons are of three types: major, microtubules, microfilaments, and intermediate filaments. And in this, about the micro, and that is actin filaments are there mostly. And if you come to the cell organelles, there are membrane bound organelles and there are non-membranous organelles. Non-membranous organelles are ribosome, centrosome, basal granules, and also nowadays there are a lot of spheres full with microRNA. That is one type is coming. It will come to the textbook as well in within couple of years. Then if you come to the membrane bound organelles, you know endoplasmic reticle, Golgi complex, etc. You all know these. Those who don't know, just go for your textbook. Now generic cells, 95% of cellular membrane is intracellular organelles. 95%. If you look into these, now membrane composition, this typically looks like that. Different kinds of proteins within the membrane. Now lipids structure, I will give you the glimpses that in this lecture I thought about the molecular logics of life. So membranes, lipids are there, you know, without lipid there will be no membrane formation. Without membrane there is no cell. And you see within the cell the endomembrane systems are there, that is endoplasmic reticulum, mitochondria, Golgi apparatus, and various others. So the structure, the basic things of lipids, generally if you analyze a lipid molecule from cell membrane, you will find that skeleton of the lipid molecule is glycerol. You know the glycerol structure. Then 3 fatty acids, when they form, that is exclusion of 3 water molecules, it giving you triglycerides. You know, alcohol plus acids, water plus hello, no chemistry student? Ester plus water, okay? So this triglyceride, triester. Now if we look into these, you see in a typically membrane lipid, you will find a head group. The two of the weight of glycerol is linked with 2 of the long chain fatty acids. These are known as fatty acids. And another OH is linked with the phosphate group, and that is as a whole this part is known as phosphatidate. This phosphate group is further linked with different attachments. Here it is choline, that's why this is known as phosphatidylcholine. This part is long hydrophobic chain is there, and this part, the head group, we call hydrophilic. So this gives a polarity to the lipid molecules. So these I took from the class notes. That's why this is your turn. I'm not go for that one. Now, three membrane lipid types. Phospholipids, as I discussed, phosphatidylcholine. Then if it is head group is serine, phosphatidylserine. If it is inositol, then phosphatidylinositol, and so on. This is the cholesterol molecule, and this is different kinds of glycolipids. Lipids, when they are linked with sugars, that's a glycose. Sugars, then those are known as glycolipids. These are present in cell wall, in cell membrane, plasma membrane, elsewhere. Now, if you look at the phospholipids, phosphatidylcholine, choline head group, I have shown you. Just simple serine, if it is linked with the phosphate, this is known as phosphatidylserine, amino acid serine. Then phosphatidylethanolamine, if ethanolamine. Ethanol, you know the structure. The NH2 group when linked, that is known as ethanolamine. When it will be linked with the phosphate group of that phosphatidate, then you will get the phosphatidylethanolamine. There are also sphingomyelin. When serine replaces glycerol backbone, it is known as sphingomyelin. One part is glycerol is there, then in the serine residue in the phosphate, if it is removed by the glycerol backbone, then it will be the sphingomyelin. Phosphatidylinositol, one of the less common types, it is mainly involved in various signaling, inositol signaling pathways. Then hydrophobic, soluble part, this is and hydrophobic part. This is the cholesterol structure. Cholesterol, this is known as cyclopyridophenanthrene. Hydroxyl group is very small polar, OH group is there, and this gives the polarity, the head and tail part. This is flexible hydrocarbon chain, tail part, and this is the rigid. If you look at the structure of lipid, you look at this kink generates from unsaturated fatty acids, and if you look, these two lipid molecules are side by side, there is a gap because these steric hindrance. A cholesterol fits oil in this gap. Now glycolipids, when sugar moiety are attached to the lipids, sugar residue. This is serine, when it is attached to this is known as glycolipids. Now cell membranes, when lipid, they have the polarity. So these chains are here shortly shown. These are lipid bilayer. Outer membrane of cell that controls movement in and out of the cell. These are made of phospholipid bilayers. Structure, and occasionally there are a lot of proteins, that there are pores, there are channels, and various others for the permeability and different types of functions of the cells. Now, if you come to the nucleus, you see there are nucleolus. I already discussed about that. Different chromosomes, they are at different sites. What is the main function of nucleolus? Any one of you know? Synthesis of ribosome. Ribosome is synthesized in the nucleolus. Then ribosomes come out to the cytosol, then functions for translation, that is protein. It is the control center of the cell and direct cell activities. No doubt, because the programming materials are here, DNA is here, contains genetic material, that is DNA, surrounded by a double membrane, that is also true. Usually the easiest organelle to see under microscope because of its size. Nucleus is the biggest size. Usually the easiest organelle to see under microscope, that is the reason. And in most of the cases, it should be one per cell. If there is more, that is exception to the reality. The hydrocarbons, hydrated carbons, these are carbohydrates, monosaccharide, disaccharide, oligosaccharide, polysaccharides. Our business is mainly with hexoses and pentases. Pentases, they are ribose sugars. When you talk about the DNA and RNA, deoxyribose, that is ribose sugar is there, a base is there, and phosphate is there. We will come to that point. Then disaccharide, two monosaccharide molecule, when they are united by glycosidyl linkage, this is disaccharide, oligosaccharides, and so on. So this is monosaccharide, aldose and ketose. Fructose is a ketose, you know all these things. And when they unite, you see cellulose. All of you know cellulose, a major constituent of plant cell wall. consist of longer linear chains of glucose with beta 1,4 linkage. You know what is alpha and beta? You put it, this is our projection formula. If OH is up, then it is OH is down, it is alpha, and OH is up, it is beta. And you see one carbon atom, 2, 3, 4, 5, and the sixth one is here. So the linkage is here. So this is the cellulose structure in general. Now if you go to the Botany online website, you will find all the schematic arrangement of cellulose chains on a microfibril in plant cell wall. Another is that every other glucose is flipped over due to beta linkages. Now amino acids, you know, structure of amino acids here, NH2 group and COH, organic acid group, and here is different, H and R group. If R is H, it is glycine, R is equal to CH3, a mistake is there, CH3, it is known as alanine, and so on. Is it okay? Are you enjoying, or I can come directly to the epigenetics?
No, sir, it's fine.
Sampa, hello?
Yes, sir, we are enjoying. You may continue.
Okay. So now, if you see the amino acid structure is this, and then peptide bond formation. Peptide and peptide bond. A peptide bond, if you look, the amino acid structure here, NH3 group, another amino acid here, NH3 group. Now this is COO minus. It will react, a water molecule, one water molecule will be excluded, and you will get the structure like this one. C double bond O, single bond N, H. You know, N is more electronegative, it can pull out the bond from hydrogen to here, and they can share here. Oxygen will pull out the oxygen to this part, and then partial double bond character it gives. That's why it has been shown. You can, in the next slide, it will be clear. So this is the peptide bond. Peptides can be extended by adding multiple amino acids. So multiple peptide bonds in a sequential order. So if it is 2, then dipeptide. If it is 3, tripeptide, and so on. Oligopeptides and polypeptides. And large chain polypeptides, when they are uniformly folded to give it specific folding pattern, then those proteins are active and functional. And about these, there are two types: major structural proteins and other is enzymatic functions, you know. So biologically active peptides, you have heard about lot of times about glutathione. So this is a glutamic acid, and thiol group is there. This is cysteine. It is actually 2 peptide bonds are there involving 3 amino acids. So glutamic acid, cysteine, and this is glycine. So that is glutathione. It works within the system with the help of glutathione peroxidase. There is an enzyme. It scavenges many free radicals. Now, what do proteins do? Protein have different kinds of functions: regulation, movement, signaling, catalytic function, enzymatic functions. Then in immunology, you know, immunoglobulin, there are a lot of transport, sodium potassium transport pump. Signaling, you know, RAS map ke pathway, those who are reading cell biology, you know very well. Then catalytic enzymatic function, all of you know. And if you go to the membrane system, muscle system, when you are running, the muscle movement, most important. So various kinds of functions among the proteins that they are controlling the life processes. I mean, when the living organism is multi-organ system, like our human being or elephant or whatever you think. So you know the ribosome, there are 2 parts, small part and high part. Ribosome is synthesized in nucleolus, then matured also over there, then come it back again to cytosol. RNA is synthesized in the nucleus. RNA is coming. In some cases, most of the cases in eukaryotes, RNA needs to be processed. That is, initial after synthesis, it is known as pre-RNA, and after that, when it is processed, generally it is known as splicing process. We get the mature RNA, messenger RNA. Messenger RNA binds in the ribosome with the help of various ribosomal RNA, the mechanical support one after another, and you know the genetic code is read in triplet. It will be tuned with the help of transfer RNA. One amino acids corresponding to the genetic code will be brought, and you will get the polypeptide synthesis in general. Now, if we look into the protein, is it a protein? This is the cytosolic part. This is the extracellular part. If you consider, this is your membrane bilayer, a transmembrane alpha helix. Consider here. Proteoglycans are glycosaminoglycans that are covalently linked to serine residues of specific core proteins. So this is a transmembrane. Why? Trans matlab dono side, both the sides it is present. That's why it is not transmembrane alpha helix, and that is, it matches with the hydrophobic core of the membrane systems. And then core protein, it is heparan sulfate glycosaminoglycan. So when it is modified, this red part is carbohydrate molecules, proteoglycans or glycosaminoglycans. You'll find. Then we are coming to the human genome. Can you see full screen? Animation is here, that's why I am showing. Can you see?
Yes, sir.
So the chromosomes are knocking. Human karyotype, 46 chromosomes are there. Out of these 22 pairs, that is 44 are autosome, and 2 X and Y or XS are the sex chromosome. This slide you can see.
Yes, sir. Yes, sir.
A cell has the same genome, but look and work different. If you consider, just consider a human body, in your liver cell, kidney cell, lung cell, elsewhere, the genomic composition is same, but architecture is different, and it is dictated by the expression pattern of the gene. Total human genome, in the total human genome so far, the scientists have discovered that approximately 30 to 35,000 genes are there. Out of these, we are getting about 55 to 60,000 proteins. So whatever you will read initially in your microbiology classes or biochemistry classes, you will read, Dr. Devnath will teach you initially that one gene, one protein. And again, when you will come to the consecutive semesters, Dr. Devnath will tell you again, now the concept is changing. In this lecture, I am giving you the idea, because why it is one gene, one protein. No, then if 35,000 genes are there, how we are getting 55 to 60,000 proteins? One gene can give more than one protein, and there are mechanisms that it, you know, the nature of the mRNA, you know, there are intron and exon. Exons are coding regions, introns are non-coding regions. Introns are excluded. In some cases, some of the exons are also excluded, depending on the signal and machinery involved within the process of the processing of those RNA. So in that way, different kinds of, a single gene can give different kinds of 2, 3, 4 proteins, or its isomer. So one of the sperm cell will be winner, single cell, egg cell. There are a lot of food storage, nucleic acids, RNA, all these stored here. So a single cell, then it will be splitting. After matching with these, it will be splitting. Can you see this one?
Yes, sir.
What is written? Zygote written है, zygote na?
Sir, no, slide number 38 is visible.
What is written? You tell. Zygote लिखा है ना?
No, sir. Human karyotype 46
6
chromosomes.
chromosomes.
Oh my God.
Actually,
38.
the slide haven't moved into 39th.
Okay, got it. Here you are, the human karyotype.
Yes, sir.
Yes, yes,
Now you see this slide.
yes,
Yes, sir.
लिखा है liver cell, kidney cell, lung cell.
Yes, sir.
So all cells have the same genome but look and work different. How? Because of the differential gene expression. In our genome, there are 35,000 set of genes. Total chromosome element we know 46 pair, and we know that particularly these the biasness of gene, our differential gene expression comes out with different mechanism nowadays, what we call the epigenetics. That I will come just now after couple of slides. So here if we look, you can see the full form. The zygote is written here. Is it moving?
yes. This slide is visible.
here लिखा है zygote, ना?
Yes,
Yes, sir.
Okay, very good. Now full screen visible?
yes,
Okay. Rupam, I will talk to you. I am in a lecture. I will একটা ওয়েবাইনার লেকচারের মধ্যে আছে, আমি লেকচার দিচ্ছি নিজে। আমি তোমাকে রাত্রির দিকে ফোন করছি। Okay, now if you look, a zygote is the product of the fusion of two haploid cells, an egg and a sperm. Then it comes to blastula, then it comes to morula and blastocyst. Here I drawn it 8 cell stage. If you think in that way, the 8 cell stage is a period in embryonic development when the conceptus has undergone 3 cleavages. First one zygote, then single division gives you 2, second division gives you 4, and third division gives you 8 cells. Generally it is not division. You see the egg size is huge but number of cells is increasing. In molecular biology term we call the splitting of cells with the core nuclear elements, I mean the genetic elements. Now blastocyst, in the blastocyst, the mammalian conceptus in the post morula stage is known as the, this is morula. After the morula, blastula is post morula stage, and then blastocyst we are getting. Now stem cells have the capability, you have heard the stem cells. In my lab we are working with cancer stem cells, and occasionally when we compare with these, generally reviewers ask that you have to give some data on the stem cells as well, original. So for that we use a lot of cell culture. So stem cells have the capability to self-renew indefinitely and have the capacity to divide with at least one daughter retaining the phenotype of the mother. That is the definition, stem cell. Now just, you know, totipotent cells, it can give any kind. Then pluripotent, some kinds it can give, and then unipotent, particularly that cell will give only, say if it is from lung. The most important feature is that just after fertilization, if we look into the genomic analysis, if we analyze the DNA, you will see the paternal part is drastically demethylated. What is methylation? In every genome, adenine, cytosine, thymine, the bases, at certain point at NH2 group or OH group, they are modified according to the availability and circumstances within the nucleus or when they are prepared. But all these processes, modifications, are post-replicative. Whatever happening to DNA changes, basic concept is that the 4 type of bases within the DNA, they generally remain same. If it is changed, that is known as mutation. Whatever changes implied just after DNA, the main sequence will be remain, but some of the modification can be changed. Okay, so just you, it like an idol. From the soil you can put, if you put 4 hands and different weapons, you can think that goddess Lakshmi or goddess Kali. If you put 10 hands, you generally tell that it is goddess Durga. At different points and different sections of the genome, we find different modifications within that one. Some are silencing and some are dynamic. So what is that? In the concept of your genome, you know all of that heterochromatin and euchromatin. Heterochromatin from your typical cell biology, I mean cytogenetics classes, you will learn what? You will learn that the heterogeneous sorry, heterochromatin part which takes more stem and it is very dense, and the euchromatic part it takes less of the stem, that is you can eosin staining or various other staining procedure is there which binds to dendrite orange etc. which binds to DNA, then you will get deep, and in that way when you will find that generally compactness is there in the heterogenetic part. How it happens? Now initially in those days it was not known that much, particularly when scientists discovered that part, genetic, I mean DNA double helical structure also was not known in that way, but we have now progressed too much. So now DNA methylation, particularly any of these bases in the adenine, cytosine, guanine, thymine of DNA, methylation can happen at different position. But when it is happening is this fifth carbon of cytosine, fifth carbon of cytosine, that is known as cytosine methylation, and in general in epigenetics we term it as DNA methylation. Okay, now here you see this genetic architecture slide. See, DNA double helix is clear?
Yes, sir.
So DNA double helix, if you consider the diameter 2 nanometer only, then it is wrapped up nucleosome core of 8 histone molecules. You know histone molecules H3, H4, H2, H2B. Each of these 4 types gives 2. That's why it is 8. And this compact octamer, another histone is H1, this one. This is known as also linker histone. Then it forms the nucleosome, we know. Histone H1 tail, and you know this is overall nucleosome is 11 nanometer. And then it makes a compact structure of 30 nanometer, then it gives 300 nanometer, ultimately wind up to 700 nanometer, whatever we observe. And then this is your chromosome. You can see the animation, na? Epigenetic architecture,
No,
can you
sir.
see?
No, sir. There is no animation.
Yes, sir.
Sir,
Yes
we
or
can
no?
see only slide. Sir, we can see only the slide.
Blank slide. Now full slide, can you see?
No, sir.
This you can see.
Yes, sir.
You are
Yes,
on
sir.
that slide, zygote and that one, only that one.
No, sir. The
No,
next slide. We can't see the animation,
sir.
sir.
We are on
Acha, acha. Now you can see the full slide, genetic architecture.
Yes, sir.
Okay, so here you see the DNA double helix 2 nanometer diameter. Then, as I told that 4 types of histones H2A, H2B, H3 and H4 gives 2 molecules. So 2 into 4 8, that is octamer, histone octamer. Your DNA is wrapping this histone octamer 1.7 times, and if this octamer it looks like a cricket ball structure, the nucleosome. And this yellow part is histone H1. This is also known as linker histone. It protects the barrier to mix up and to be digested. The rest part with the nucleases, that is mostly DNAs. And then it is when these are compacting with other associative factors and proteins, it is giving 30 nanometer chromonimo fiber. Then it is giving a 300 nanometer fiber or 250 nanometer wide fiber and 700 nanometer ultimately, and it is ultimately wrapped into your chromosome, that is known as 1400 nanometer fiber. So if you look into the cells, as I was discussing, that your different or lung cell or liver cell or others, they have different kinds of architecture. So same with same genetic setup, but there is different epigenetic setups. As we are talking about cytosine of adenine guanine cytosine, cytosine methylation at carbon 5 position of cytosine, or histone lysine acetylation, methylation, etc. This is known as epigenetic architecture. So what I was, then there are other modifications, a peptide like ubiquitin, or there are in the serine position there are phosphorylation, you know. Now the two main components of the epigenetic codes are DNA methylation at different cytosine, and there is histone modification of different histone tails. Histone proteins, although it is octamer, but it's N chain and C chain, that is known as tail. N terminal tail and C terminal tail are little bit exposed, and from generally when we generally number from COH group, but for the histone proteins we count it number 1 from NH2 terminal. So if we look into the histone 3, there is in the 4th position there is a lysine. Lysine can be acetylated or methylated, and in that way it gives a dynamic feature architecture of the chromosome surface, and different kinds of other enzymes and proteins which have the capability to read this mark, they will bind, and then it will be available for further, like you know, transcription, otherwise not. So this modifications type which triggers the gene expression with the help of different transcription factors, these triggering molecules and the modifications along with all these things, these are epigenetic phenomena which dictates the expression pattern of particular gene in a particular tissue, and that is molecular epigenetics in current days. By definition, if you want to learn, sir, what is epigenetics? Epigenetics is beyond genetics. Above all, which generally guides or as a prophet to tell the gene which part of the genome or which gene will be expressed. That is dictated by this all kind of modifications or epigenetic modifications, or altogether these are also known as epigenetic code. A lot of functions, a lot of amino acid side chains are there, different modifications are there. You just keep it in mind that all these phenomena is not essential for same gene. Depending on the position of the gene in the chromosome and in which chromosome number it is there, the signaling pattern and the modification pattern is essentially differing, and it dictates the expression status of that particular gene. Now, how epigenetic control? Epigenetic control is important throughout the development and goes wrong in disease. So if you look at the fertility program, as I already told, if you drastically analyze the nucleic acids just after fertilization, paternal part are drastically demethylated, whereas maternal part remains intact. So it is natural that some kind of surface binding, the chromatin surface binding proteins are there, which is protecting the maternal DNA from demethylation. But that is present in the egg side, because if it is present in the sperm side, then originally before fertilization the sperm DNA would be demethylated, but it is not happening. Just after fertilization it is demethylated. So demethylase enzyme is coming from the egg part, but it is egg chromatin is protected for demethylation due to the presence of other protein on the surface of the chromatin of the maternal part. This is general, and in this way scientists starts their research activity. When you find a new protein within a complex, you will you can tell this is the component of that complex. How we do? We generally do the immunoprecipitation. Just take one antibody against any of the protein, then analyze by Western blotting. Your professor Dr. Devnath will explain you what is Western blotting. By Western blotting, you can resolve the protein. If you get number of proteins, you can tell that is a complex part of that particular protein against which you use the antibody. So now if you look in the blood cell development here, or mid gestation embryo, or any kind of things, different types of proteins are involved. Now already when I started 8 cell stage, then it comes to 16 cell stage, and compactness is coming with the expression of proteins like E-cadherin, the cell-cell junction protein. Two cells are attached by this E-cadherin, CD44, different kinds of cell-cell attachment protein that makes the tissue integrity. Two cells must be attached to make a tissue. I mean different cells. So when two cells are essentially attached, these are attached with different kinds of surface active protein. These are known as membrane surface proteins or cell-cell binding protein like E-cadherin, CD44 and various others. So now this compactness when happens, we will see different changes, and we will see different 3 layers: ectoderm, mesoderm and endoderm. It is developing just after, and then we, from these 3 layers we will get our organelles, different kinds of organelles, organs. So this is in general in germ cell it happens, and in tumor cells any kind of deviations from these modifications will give importance to one gene which is not essential that part to express the particular protein, and cells will indefinitely grow, and that is the major cause of tumor development in any cases. It could be from the development, it could be after development, like you observed in many cases of tumors and cancer progression. That is the mechanism. Now whatever we got, if you go back again, the reductionist approach, these are the building blocks of adenine, guanine, cytosine, thymine, that is deoxyribose sugar, this is phosphate, and we are getting here the base deoxyadenine monophosphate. Then if it is triphosphate, then DATP, it is monophosphate, diphosphate and triphosphate. Now if you look into these in the DNA sequences, when C cytosine with the help of an enzyme DNA methyl transferase, we abbreviate as DNMT, or in microbial system it is known as DMTS, methyl transferases. So cytosine 5 carbon is there, high methyl cytosine, this is cytosine 5 position. So now you imagine within the DNA sequence it is, so it is DNA methylation. We generally are marked like a lollipop, blank lollipop, it is unmethylated region of cytosine, and when these cytosines are linked with CG with the phosphate, sugar phosphate backbone, so this is known as CPG island. So genes are there, particularly in the promoter regions of genes, these are heavily methylated, and other types of proteins when bind, they block the transcription factors to bind, and RNA polymerase cannot further process the transcription. In that way, DNA methylation blocks the gene expression. When it is methylated, it drags other, it is hydrated and it drags other type of proteins when it is bulkier. Crystallographer view. Now here if we look, if I do animation you will not see, so I am showing you step by step. It is a cytosine from your DNA, this is your DNA chain, just to show you the molecular level. This is cytosine 5 carbon position, S-adenosyl methionine is the donor of the methyl group. It is converted to SA, that is known as S-adenosyl homocysteine. DNA methyl transferase helps the process, and this is the methyl group attached, as I saw you in the crystallographer's view here. So this is methyl group, it will be hydrated, bulkier, and then this position will be covered with a type of protein known as methylated DNA binding protein, that is MBD, methylated binding protein within the DNA. Now see, when they bind, they have more affinity towards this side, then transcription factor and RNA polymerase, they will be blocked, they will not be able to synthesize. So now we are coming to the histone modifications. methyl switch of histone, their complex methylation is there. It could be, if we look into the lysine, it could be acetylated, it could be monomethylated, dimethylated, trimethylated. Lysine can be ubiquitinated. There is also SUMO group, a small peptide group or large peptide group. These are ubiquitin and SUMO. So they can be methylated, and when they are methylated, acetylated, or ubiquitinated or SUMOylated, after that, when they are in the chromatin surface, the reader protein will come and bind, and they will accordingly, the transcription factor will also come and bind. Accordingly, they will kickstart the activation of transcription. So here, if we look histone 3, 1, 2, 3, 4 lysine is there, at 9 position there is a lysine. If 4 position it is methylated, then it is active, but in the 9 position if it is methylated, then it is inactive. In 9 position it should be acetylated. If it is methyl group is there, it will prevent the S10 phosphorylation. If there is S10 phosphorylation, there is no methylation in K9, it is acetylated, then phosphate group will kickstart here, lysine methylation and transcriptional activation of the gene. Nucleosome structure, I have shown you. In the nucleosome structure position, those histones are there. We are talking about that part, not overall. In the promoter region, nucleosomes. Now, competition for combatant modification enzymes. There are different enzymes. It will be too much for you guys. You just keep it in mind. Depending on the signals and the microenvironment within the within a cell in the nucleus, the different kinds of different parts of the histone proteins, they will be modified by different enzymes. It is signal dependent, and there is a much emerging concept that nucleoskeleton and cytoskeleton, when in the membrane surface is something happening, it will be felt in the nucleus through cytoskeleton. You know, 3 cytoskeletons I told you: actin, microfilaments, microtubules. These are linked with the nucleoskeleton through the nuclear membrane proteins, outer membrane, inner membrane. Lot of proteins are there. Like these are mechanically connected, and when there is a change in the membrane surface, any kind of mechanical stress, when a receptor is taking care of a ligand receptor complex, this is the most primitive, earlier demonstrated mechanism that signal will be passed. When a receptor is bound to its cognate ligand, there will be phosphorylated autophosphorylation, then RAS-MAPK pathway or various other signaling pathway will send cascade of signaling to the nucleus, then gene will be expressed. That is the premier concept. Along with this one, when the ligand is binding in the surface of the membrane, the protein, particular receptor, it will be filled with the inside cytoskeleton, and it will be transmitting that mechanical stress to the nucleus through this is known as linker of nucleoskeleton with cytoskeleton, that is known as link complexes. And it is nowadays emerging that mechanotransduction. So cell will realize first immediately when there will be any change. Like in your door, you are sleeping in your home, in your bed is the nucleus you consider, but door there is a knock, the sound will go, but mechanical transmission will be go through the bricks and or soil, whatever is there, or your roof, if it is wooden structure, generally it will be felt definitely. But depending on the strength of the knock or signal, the amount or extent of mechanical transmission depends. So if it is enough, it will also activate the genome for transcription. Now reaction speeds, you know, chemical and biological reaction, there are kinetics, etc. I will not take much time. If you even grow a E. coli cells, you make a kinetics when it is growing. A E. coli cell takes typically 20 minutes for duplication. A human cell takes 24 hours. So speed kinetics you can do. Enzyme reactions you can measure the rate. Chemical kinetics, different chemical reaction. If you can put the product versus time, linear plot will come. But in enzymes, you will not get the linear, complicated process. The generally it comes in this way. The product time. Next, if you look at Michaelis-Menten equations, you will get this type. Interpretation of the set. If it is low substrate, in a reaction there is an enzyme, there is a substrate in enzyme kinetics. So rate is very dependent on substrate. Binding rate is limiting. So substrate plus enzyme, you will give a transition complex, enzyme substrate complex, then product plus your enzyme will be free. If substrate quantity is high, rate dependent, saturation of enzyme and chemistry rate is limiting. So here typically we got V0, that is velocity of the reaction, and substrate concentration. Generally we plot, we will get this one. In this way, Michaelis and Menten, they deduce this equation, Vg equal to Vmax into S divided by Km plus S, known as Michaelis and Menten equation. So why I showed you, how you will tell that this enzyme is involved in cancer. So take the cancer tissue, measure the particular enzyme, we measured it, particularly DNA methyl transferase, the CH3 donor is SAM, as I already told you. Now the efficiency of that enzyme you can measure, high expression, high activity, high expression, here in this way. So in prostate cancer I measured initially when I was in United States, in the University of California, San Francisco. I started cancer research there. Now I am continuing for the last 22 years. So we took cells or tissues, benign prostatic hyperplasia and prostate cancer tissue. You see the enzyme activity is high. So when you are finding that enzyme activity is high, you can tell this enzyme may have some roles in this cancer. So if you inactivate that enzyme, you will get some medicine and you can control the cancer. That is the general view. That's why I'm showing you. So in different way, that there are medicine, if you inactivate, say the typical solution for inactivating the DNA methyl transferase enzyme is use of 5-azadeoxycin. In cancer cells, they generally rapidly grow and they cannot identify difference between cytosine and this azacytosine. Instead of carbon, there is nitrogen. That's why it is known as azacytosine. 5-aza-2'-deoxycytidine. If you take instead of CTP, if you take more, CTP will also be there. If you put only azacitidine, cells will not grow. Now, enzymatic functions, it will be too much high for you. You analyze the structure of the enzyme, then you typically measure the, we did it, the reaction mechanism. Then you do some bioinformatics works, what is happening around the globe, what information is there and what you want to know, new one. So in this way you just analyze different softwares, tools and information centers are there. You click there, you will get all these things, predict the CpG island, then you prepare your protocol, you do the experiments and assess quickly. So we did something, say clustering gene. It is a gene which is functioning, secretory clustering and nuclear clustering there. Lot of cancer studies we have done from here involving with this enzyme and DNA methyl transferases. So here is a CpG island is there within this, so it could be methylated and it is repressed. And in fact, it is happening by experimental evidences. We have shown various other groups are also working with different enzymes. So only CpG island of clustering promoter, it is showing this is more functional. So we measured, and how you will do, you block this part with the enzymatic functions. You can block the CpG by utilizing azacitidine. Less function, you can measure the proteins and detect the at the protein level, and you also digest the DNA and analyze by modification. You can detect the methyl cytosines. So how much extent of methylation is there, you can detect. So here if you look the methylation of high amount of DNA, you do it in vitro, then you measure and then map it. After mapping with your own data from your analysis of tissue samples, you again measure, you will get. This is the chemical mechanism how we shift. When cytosine is methylated and then NH2 group is, that is oxidative deamination we call, replaced by O group, then it is completely transferred from cytosine to uracil, cytosine to thymine. Now this is hotspot for mutation, because when it is spontaneously deaminated or oxidative deamination we call, in H2 group was there, now it is oxygen, now it is methylated, now it mimics, instead of cytosine methylated cytosine, when it is transferred it is give you thymine, then thymine in the DNA it will completely turn into the, in case of C it is G, in case of thymine it is A, it will come. You will ultimately in the next generation you will get a mutation. So you can measure in this way, ATGC sequencing and sequencing pattern. A is generally it is showing there is no methylation, B pattern is asterisk is there, change according to those changes we can say that partial methylation is there, and complete methylation, high peak signal, it is telling us that a lot of methylations is there. So there is, you will learn that small RNAs are there. This microRNA research is ongoing in my lab also. It will be hard for you guys. I am not going too much with this one. Now I will close. There are a lot of immunofluorescence. Then you will get the confocal microscopy and all these things. Coming to the point what I discussed today, any one of you, my dear students, if you want to explore your endeavor, scientific endeavor in research, you don't think too much. And when I entered into my PhD courses, I read fast. I entered in 1991. After passing, there was a gap due to my ill health. And after 90 to 91, jaundice আমাকে কাবু করে রেখেছিল। তারপরে জয়েন করি প্রফেসর এম কে পলের ল্যাবরেটরিতে, আমাদের কল্যাণ ইউনিভার্সিটি বায়োকেমিস্ট্রি বায়োফিজিক্স ডিপার্টমেন্টে। I first encountered this prefatory chapter, it is known Annual Review of Biochemistry. As a biochemist, I love this scientist, Oliver H. Lowry. You know Lowry method for protein estimation. Nobel Prize many scientists got, but no scientist have higher citation than this scientist paper, particularly protein estimation. So he wrote the prefatory chapter in that way, how to succeed in research without being a genius. What he did in his research in this paper, particularly he was trying to, in those days the protein estimation took, I mean, more than 24 hours, 1.5 day. So he reduced the time. In his time it bring it to half an hour, and nowadays in 10 minutes or 5 minutes we can get Bradford method, exploiting the Lowry Bradford again developed. And more than it is, I am talking about around 1999, an article came that by that time it got more than 1 lakh citations. No paper, even DNA double helical structure has got that much citation. So just simply think, and my view is that after working more than 35 years in research field, if you have a test tube, you have acids, bases, carbohydrates, lipids, proteins and DNA, and only a spectrophotometer, you can start research. Mix them, take OD, mix them one with another, one with 2 others, with 3 others, and then all. You are close to real life biomolecules. That is my message to my dear students who are, and my dear colleagues also. So I will show you the sum of the, I worked with this guy in Canada. I was an assistant professor grade, but in Canada you have to be there with a prophet like professor, and this man is very kind and loving to me. That's why he come to my laboratory in 2016. I finished work with him in 2004 and I come back. He insisted me to stay there and get a regular position at the rank of associate professor by couple of years. But I was, I mean, too much stubborn to come back to my country. I never opted to stay in outside. So I visited so many countries, you know, but I opted to come back, and when I settle all these things in my laboratory, he saw the papers in publications and then contacted me. Samir, so you have established your own lab? Yes, sir. So when you were inviting me to visit, I would say any day you come. I invited him in 2010, 2015, but ultimately he came in a workshop of epigenetics I arranged here in this institute. He came as the chief scientist to deliver lectures on that. Some of the participants here and some of the lab mates is here. My colleague here, myself and Professor Mosejid, and in my office from where I am delivering lectures. He was there that is in 2016. You see, when I was in San Francisco, this is I cherish these photographs when I was in San Francisco during 1999 to 2002, 3 years, all over 3 years. This is, this is the culprit at a young age at that time. My wife Aditi, she was also working there. She is a veterinary officer in serving for the West Bengal government. So my family is split. I am in Rourkela and they are staying in Kalyan. My 2 kids. And this is our mentor, my mentor in University of California, San Francisco. And this you see the technicians and all the students from Brazil, this is American, this is from Japan, this is from Taiwan, Chinese, and various other countries. So all were not there in that day. This is Indian from South India, a doctor. She was there also at that time. 25 members we were, but if you had attended, this is in an Italian restaurant to farewell one of our guy. He took the photograph. He is not here. I will show. No, this is here. He set the camera. This is Long Chain Lee, a genius. I've seen he's a doctor, but computer and software, easy. It is water like for him, and he generally inspired all these things, bioinformatics searches, etc. in Professor Rajbir Dahiya's laboratory. He is India. That is University of California, San Francisco chapter for 3 years. He also insisted that you stay here in my lab and eventually you will be a professor. I did not opt it. I went to Canada, enjoyed there. So this is Rajbir there. I am presenting one of the paper in AACR, that is American Association for Cancer Research. Every year, that is annual meeting. Last 2 years he is hampered due to COVID. So I was there for 3 years. So 2001. In 2000, there was no sufficient data for my presentation, but in 2001 and 2002, I presented. I attended 2000 also, but at that time, sufficient data from my hand was not there. And now this is memorable moment here. Says in 2000, perhaps it is 2018 in molecular biology classes, from my dear students here in NIT Rourkela, and that was 11th April. And I was taking 2 consecutive classes. When I asked them that, do you need a break? They told, yes sir, we need a break. Give us 10 minutes break instead of 5 minutes. Why? No, we need it. Then within 10 minutes they arranged. That was my birthday, official birthday. So you see the crowd and their enjoyment. It was huge. A professor, what we want from students? We don't want more than this. The cheerful guys, and they all are brilliant. They passed well. They are placed elsewhere. And in the 2020-21, I don't have photograph. You know the reason, that is due to COVID. Culprit. So current strength of the laboratory, here you see 8 students are working. Priyanka Saha, Bengali girl, she is now assistant professor. Just left before COVID, joined there as assistant professor. Now work is hampering. By that time, if it is not COVID era, she would have submitted her thesis. Then Kirtano, she is CSR, very brilliant student, working hard. Niharika, she is working in a ACRB project in my lab. Dr. Mr. Somen Manna, DBT project, now Institute Fellows, Convert DBT project is end. Ankon same. Suraj Pratap Singh, he is from UP. Somnath Mandal, another guy, is qualified net. And as a JRF working, Tirthankar Paral is an ODA student. He is working also at UGC net qualified. And unlike Kolkata University or various other universities, our institute is generous to give a support of technician. That is, she is an institute employee, but dedicated to help my laboratory, practical classes and maintenance of the laboratory. She is very dedicated fellow, work hard. And these are some of the students. Due to COVID, their shifting duty is there. I took just photograph this today. These 5 students are working, others are coming at night or various other times for COVID management, crisis management. Some of the alumni of the laboratory, my first PhD student Monmun Dev, she is currently working in Edinburgh via Texas and Oxford. After doing my PhD, she was in Oxford for 1 year, then moved to Texas. And again with an Newton Fellowship, she went back to Edinburgh, the birthplace of epigenetic studies, Conrad Hall Waddington. So that is in during 1940s. Dipto Sengupta, while they were working in my lab, he joined just after 1 year of Munmun, but they fall in love during work. They are married. He is also in Edinburgh. He went in 2017 first with Newton Fellowship, while Munmun was in Texas. This is Samsi, the most brilliant student, first best student of NIT Rourkela, Life Science. She did PhD under my guidance, produced lot of review articles and research papers. Very bright. He was enjoying the National Postdoctoral Fellowship at ISR Bhopal, but due to family reasons, he stopped. I don't know what is the reason. Arunima Silpi, she is currently in NIH. The first bioinformatician joined my laboratory in 2011 and left my laboratory in 2016. Now she is, just few days back, she wrote me, sir, I am applying for the staff scientist position. That gave me enormous pleasure. Staff scientist position in NIH, National Institute of Health, the most prestigious, supportive, organization of United States, and you can count also worldwide. In Germany there is Humboldt, in Japan there is JSPS, also similar kinds. Sabnam Parmi is working in University of Göttingen, Germany. My first postdoc, Madhumita, she joined in 2012, worked and learned many things because she was microbiology background from MIT fellow. She was not that much skilled. Then she joined in the engineering college at biotechnology department in Bankura, Bankura Unnayani Institute of Engineering, like that name, UIE. And various others like this. She also got PhD. She was a woman scientist. These two guys were in the DBT project, but I am very sorry to say that they were not able to produce any data. So after 2 years, they themselves, I never kicked out any students, my dear students. I asked them repeatedly, stay, work hard, you will be such. They told, no sir, we are not getting. So I asked them not to write their failures. I asked them show family reason. So both one showed that they will have to take care. He came from Malda, and this guy is from our Biochemistry Biophysics department. He is from Calcutta, but he was also not able to do. He showed his reason that mother illness. Nivedita Pradhan, she is residing here. She got in degree in 2019. This is another guy, evergreen, very jolly guy, Neeraj Nag. He was here for more than 2.5 years. From here in my lab, learned many techniques, qualified NET, and then told that, sir, my mother is ill, I'll have to go Calcutta. I never. Family first for me. If you are happy in family, you will be able to flourish in your professional job. So I immediately released him from Institute Fellowship, and he is in touch with me. So, you need a happy family for your happy profession. This is my family, my daughter. I am happily announcing here, she stood 8th in the West Bengal Board of Secondary Education. If examination is there, she was confident, she would have first or second, but unfortunately examination is not there. But I told her that yes, it is nothing wrong with that. You stood between 1 to 10. This is my dear son. He is now second year physics honors from Kolkata University. He told just after 11, Baba, I will study physics honors, no engineering, no others. Although I put him in different organizations who are giving coaching for engineering entrance, etc. But I'm not worried. But he is now doing very well in physics honors. My wife, I already told, my wife is a veterinary doctor. She is working for West Bengal government. Now posted in Kolkata. But family was in trouble during 2012 to 2016. because of a transfer to North Bengal, then in a remote place in district of Nadia, far from Krishnagar. That is, anyway, we overcome all these things, and I always pray to Goddess Saraswati. If you want to see my lab publication, you can go to this one, PubMed publications. The link is here. There is a link to the link below. And that's all. Thank you.
That was such an enlightening and a valuable presentation. Thank you, sir. Lastly, I would like to call Dr. Shudeep Samadar to put an end to this webinar with a vote of thanks.
Thank you, Shanchari. A very good evening to our honorable speaker and all the participants present here. It's my immense pleasure to welcome Professor Samir Kumar Patra to the third lecture of Biology Lecture Series 2021, organized by Department of Microbiology in collaboration with Competence Building Cell, Research and Development Center and Internal Quality Assurance Cell, VijaygarhJyotishRay College, Calcutta 32. I am also grateful to our honorable speaker, sir, for taking out time from his busy schedule to deliver this wonderful and informative lecture, which extends from the basic biochemistry to cell biology to epigenetics and cancer epigenetics. This will definitely help us in our future scientific endeavor. Thank you, sir. I would like to thank Dr. Rajyasree Niyogi, Principal VJRC, for her constant inspiration to organize such lecture series for the benefit of the students. I also congratulate our students Miss Anindita Paul and Miss Sanchari Sen Sharma for conducting this lecture in a beautiful manner. Next, I would like to congratulate Dr. Shampa Devnath, convener of this lecture, Dr. Prosenjit Das and Dr. Shilajit Borua for their constant technical support during the session, and all the other members of the organizing committee. I would also like to thank the other faculty members of the Department of Microbiology for their immense support to conduct this lecture successfully. Lastly, I would like to congratulate all the participants for their presence and their patience during the lecture. Thank you all. Thank you.
Any specific question from anyone? Okay. Thank you all.
Thank you, sir.
Thank you, sir.
So,
Thank you, sir.
Thank you, sir.
Shampa, so
Thank you.
I can leave now.
Okay.
Okay, we'll talk in our group.
Sir, for your wonderful lecture session.
Thank you very much.
Hmm.